Hello class, so this is the second part of the series on carbohydrates. And as a reminder from what's discussed on the first part of the series about disaccharides, that's when we have two monosaccharides that is joined together. And as they give an example right here, glucose, and they put alpha right here because of on the monomeric carbon, the hydroxyl group is pointing down. When it is pointing up, remember that that is beta. And then also just remember that in this particular form, this is a hemiacetal. You can also have a ketoacetal when this H group is replaced with a carbon group. And then you have a full acetal when you have that glycosidic bond, where you have an ether that's right here and the ether chain that is right here as well. This makes up a actual acetal. And if you replace this hydrogen with a carbon group, then that is a ketal. And so when you have two monosaccharides and they're joined together with condensation with a release of water, it forms this glycosidic bond, and that monomeric carbon once again is acetal. And so when it's an acetal, it no longer has this open and closing feature, so it is now a non-reducing sugar. If you go all the way to the right end, where this monomeric carbon is still in the form of a hemiacetal, this is the reducing side of the sugar. And just as a reminder, as far as two glucose being joined together, that is the name of maltose. And when you also have a binding between two different sugars, when it is pointing down like this is a alpha type of bridge, so it is 1 to 4, where it starts off with 1 at the monomeric carbon and it joins at the 4th position of the second carbohydrate. And now, in this theme of joining together monosaccharides, and when you have multiple that is joined together, you start to form a polysaccharides. And the majority of natural carbohydrates are usually found as large polymers. So these polysaccharides can be a homopolysaccharides, which has this one monomer unit that's repeated over and over, or you can have a heteropolysaccharides, which has multiple monomer units. And so other characterization of polysaccharides is they can be linear joint type of glycosidic bonds, or they can have the branch which has multiple types of the glycosidic bonds. And as far as polysaccharides, the monomer units are added and removed as needed by the organism. And so this is pretty much just more so just general education, and to start to become familiar with certain types of polysaccharides, and they're just listed here on the table for you to read on your own. I'm going to highlight more of the ones that you need to start memorizing and become really familiar with. The first type is the homopolymers of glucose, which is starch, glycogen, cellulose, and chitin. And so glycan and starch are main storage polysaccharides for energy. And as far as glycogen, this is a branched homopolysaccharides of glucose, where the monomer is formed of a 1 to 4 chain. So one of the glucose, is the monomeric carbon is joined to the 4th carbon of the second glucose monomer. And then there is a branch point where the anomeric carbon is linked to the sixth carbon of the other glucose monomer, and this linkage happens every 8 to 12 residue. And then as far as starch, it is a mixture of two homopolysaccharides of glucose, where you have the amylopectin, which is like glycogen, but the glycosidic bond of the anomeric carbon to the sixth carbon of the glucose monomer occurs every 24 to 30 residues. And then you have amylose, which is an unbranched polymer of alpha, with the glycosidic bond being a alpha 1 to 4, which is the 1 is the anomeric carbon, and the fourth carbon of the other glucose monomer. And so we're going to be working with this in lab as far as dealing with starch, and then an enzyme called amylase. See how it's very close to amylose. So this enzyme actually hydrolyzes the starch and start breaking off the glucose, and then from there we can actually detect the glucose. So this will be coming up in your lab. And so this is more so of the structures of glycogen and starch. That's why you can have a visualization of what I was talking about in the text form. And so right here you have the glycogen, where you see that it has this alpha branch of 1 to 4, but also it has this beta branching of 1 to 6. And so you can tell it's on the sixth carbon because you start counting from the anomeric carbon, which is 1, 2, 3, 4, 5, and then 6. So that's how you see that it is a alpha 1 to 6. And it's alpha because it is pointing down. And so that's how you know what you have as far as with the glycogen. Dealing with polysaccharides. But then as far as for the starch, that is listed right here at the bottom, which is just a linear chain of glucose, where it's all alpha to alpha glycosidic bonds, where it's one position of the anomeric carbon to the fourth position of the second monomer. And remember, at the very end, that is the actual reducing end. And the other ends that have these glycosidic bonds, they are non-reducing part of the sugar. And so we also have cellulose and chitin. And so with cellulose, it's a linear homopolysaccharides of glucose, where glucose monomers form a beta glycosidic bond of the anomeric carbon attached to the fourth carbon of the other glucose monomer. Also, hydrogen bonds form between adjacent monomers. There are additional hydrogen bonds between chains. Structure is now tough and water insoluble for the cellulose, and it makes up almost 50% of plant mass in cell walls. And cotton is nearly pure fibrous cellulose. And then you have chitin, which is a linear homopolysaccharides of N-acetyl glucosamine. And so it has this monomer of N-acetyl glucosamine monomers with a beta glycosidic bond with the anomeric carbon attached to the fourth carbon of the second monomer. And then this form an extended fibers that is similar to those of cellulose. This also has a characteristic of being hard insoluble and cannot be digested by vertebrates. So the structure is tough but flexible and water insoluble, and is found in cell walls, in mushrooms, and in exoskeletons of insects, spiders, crabs, and other arthropods. And so this is more so of the structure of cellulose, where you have the glucose monomers, and it has this beta linkage, and it's 1 to 4 rather than that 1 to 6 that we've seen previously for the other polysaccharides. And then as far as chitin, remember that this is something that's found on the exoskeleton of insects. This slide gets me every year because I feel very uncomfortable when I see these big old roaches. I'm not a fan of roaches. But in any case, let's continue on. So remember when I said that N-acetyl glucosamine, that is what this structure is. So you have your glucose, and now you have this N-acetyl that is attached to the glucose ring. And so that's why you have a different type of monomer, and it also has this beta glycosidic linkage where it's between 1 and 4. Now as far as having heteropolysaccharides, this is what is presented right here within an extracellular matrix. So the cellular matrix, which holds the cells together and provides a porous pathway for the diffusion of nutrients and oxygen to individual cells. The extracellular matrix is composed of an interlocked meshwork of heteropolysaccharides and fibrous proteins and other compounds. But we're going to focus on the heteropolysaccharides. And so as far as with the heteropolysaccharides, you have proteoglycans, are macromolecules of the cell surface of this extracellular matrix, in which one or more sulfonated glycosaminoglycan chains are joined covalently to a membrane protein or a secreted protein. And so here is an example of one of those glycosaminoglycan where it has a glucose ring, and then have that N-acetyl glucosamine. And the linkage is a beta glycogen, where the anomeric carbon is attached to the third carbon of the other monomer. And then you have also where you have a glucose ring attached to a N-acetyl, and then notice that this is sulfonated, and then there is a beta glycosidic bond between the anomeric carbon and the third carbon of the second monomer. And then now you have this galactose that is bound to another glucose ring that has the monomer of the N-acetyl glucosamine. You have that N-acetyl group right here, and then this is also sulfonated, just at a different position. So this is just another example of a heteropolysaccharide. And so when you have glucose, it is broken down in order to actually create energy. So glycolysis is an energy conversion pathway in most organisms. So you have glucose that goes through glycolysis to form pyruvate, and then it can go through fermentation to produce ethanol and lactate, or it goes through a oxidative state, where it's broken down to carbon dioxide and water. So glycolysis is the sequence of reaction that metabolizes one molecule of glucose to two molecules of pyruvate. This process is anaerobic, meaning that it does not require oxygen. Glycolysis is common to virtually all cells, both prokaryotes and eukaryotic. Pyruvate can be further processed anaerobically by fermentation to lactic acid, ethanol, or a variety of other final products depending on the organism. Under aerobic conditions, pyruvate can be completely oxidized to carbon dioxide, generating much more ATP, which is energy used for cells. And this is just some of the transporters enables glucose to enter and leave animal cells. This is more so just for general education, and I'll let you read this table yourself. And something I just want to give you a heads up on, because a lot of people get tripped up over the glycolysis process. So go ahead, before I even go into lecture of going through each step, to start to become familiar with these stages of glycolysis, because there are two different stages, and this is stage one, and this is the second, where you finally end up with that pyruvate, starting with glucose. But once again, I will talk about each of these steps later on in other lectures, but it is very taxing to try to remember everything. So I'm just giving you a heads up to start studying this now. And this concludes the lecture on carbohydrates.